Hello, everyone. My name is Thanaj. I am a research fellow at the University of Westminster working in image analysis, and today I'm going to present to you my work, Mass Univariate Regression Analysis for three-dimensional liver image-derived phenotypes. To begin with, magnetic resonance imaging has become the gold standard for clinical research in the study of body composition, such as measurement of abdominal subcutaneous adipose tissue, visceral adipose tissue, liver, lungs, kidneys, and spleen. Here, the UK biobank study has invited almost 100,000 participants for the medical image assessment that includes the standardized abdominal acquisition, including a 2-point Dixon sequencing, as well as a multi-echo single slice acquisition for the liver for estimating the tissue composition, including image-derived phenotypes of proton density, fat fraction, and iron concentration. Here, we made use of the liver segmentation recently published in A.I.L journal derived from Dixon MRI from 2,730 UK biobank participants to create 3D liver meshes. Our main aim is to investigate the associations between both image-derived and non-image-derived phenotypes and local liver morphology, as well as to explore visibility of statistical parametric maps in comparing groups of subjects using computational mesh-based shape analysis. We showed that 3D liver meshes are a robust method for modeling the association between anatomical, anthropometric, phenotypic variation across the liver. Here, liver segmentations were derived from the MRI of 2,730 UK biobank participants. Then image registration was performed from 20 participants and liver reference template was estimated. We then aligned the subjects to the template segmentations, and then we constructed surface meshes. Then sign distances between reference and subject meshes were computed to define 3D phenotype. We then fitted mass univariate regression analysis to compare the 3D phenotypes values from the liver meshes to image-derived phenotypes specific to the liver. Here for the template constructions, first, we performed a linear transformation using rigid and affine transformation accounting for rotation, translation, scaling, and shearing. We then applied the deformable transformation, which is defined by the vector field of displacement field for every pixel in the image. We then applied optimization, which smooths the displacement field defined by the convolution with the kernel, which in this case is the B-spline function. Here, the template construction was applied using the advanced normalization tools. Whereas no initial template is required, the template is completely derived from 20 subject segmentation images. And the first iteration is the Euclidean average image. Then the template is normalized using the B-spline symmetric normalization and cross-correlation as a similarity metric within the images. Then surface meshes were constructed using VTK across 30,000 vertices. Once the template was available, we performed a registration step where all subject-specific segmentations were rigidly and finally aligned to the template. The subject meshes were then constructed, enabling the further computation of 3D mesh-derived phenotypes. Then for each subject meshes, surface-to-surface values was measured by computing the nearest neighbor sign distances between template surface and subject surface for each vertex. In order to map the associations between a 3D phenotype and anthropometric exposure or image-derived phenotypes, we fit a linear model at each vertex, plotting the beta coefficients as a color map over the surface of the liver. We further applied threshold-free cluster enhancement, TFCE, to enhance areas of the signal that exhibit spatial contiguity. In our case, to assess the associations between surface-to-surface and anthropometric covariates, as well as liver IDPs, we performed mass univariate regression analysis adjusting for age, gender, ethnicity, body mass index, and waist-to-hip ratio. The TFCE algorithm was applied on the permuted t-statistic of 1,000 times for each analysis. Correction for multiplicity via the false discovery rate was applied for the number of vertices and the number of anthropometric covariates or IDPs tested. Here, as you can see, the model represents the surface of the liver on which standardized beta coefficients are mapped to show the regional strength of associations between here age, BMI, waist to hip ratio, adjusting for all the other covariates. Here you can see that surface-to-surface values is shown to increase with age, whereas waist to hip ratio and BMI show to decline with increased surface-to-surface values. If we take into account the Kuhn classification, we can see that BMI and waist to hip ratio are associated with most pronounced positive surface-to-surface values in the segments 3 and 6 and 4 and 7, with negative surface-to-surface values in segments 2 and 8. To determine the nature of liver modeling, we examine the effect of the liver IDPs such as iron concentration and PDFF. Liver iron was positively associated in surface-to-surface values on 36% of vertices and negatively associated on 2% of the vertices, whereas liver PDFF showed to be positively associated on 48% of the vertices. Here we see that pattern of the changes in surface values across the liver reflect the differences that might be attributed to different lobes and segments of the liver described from the Kuhn system of classification. Changes in the liver morphology associated with liver concentrations here were stronger in segments 2 and 3, while changes in liver morphology associated with liver PDFF appear to be more pronounced on segments 2 and 3, as well as 6 and 7. However, further work may allow further mapping the associations of liver cohort as well as on other organs in the abdominal cavity, such as pancreas, spleen, and kidneys. Here we showed that the pattern of changes in surface-to-surface values across the liver reflect differences that might be attributed to different lobes and segments of the liver. We further showed that increased liver fat and liver iron concentration may have an impact on the liver shape and structure. Here we show that organ shape variation could become a powerful tool for assessing global changes associated with liver damage, disease progression, and eventually treatment outcomes. However, further work to explore this in more detail may allow the further mapping of the association between genetic variations and 3D phenotypes in specific anatomical regions. Thank you for your attention.